Hi, welcome to week 3 of biotechnology. This week we are going to be really taking in all the material that we used in weeks 1 and 2 to look at specific tools that scientists use to develop new technologies. We have 6 essential questions this week. How can bacteria and viruses be used to develop biotechnology? What tools do we have to identify, transfer, and grow genes? How did the Human Genome Project and HeLa cells, those are Henrietta Lacks cells, impact the field of biotechnology? What are some potential benefits and ethical concerns surrounding the CRISPR technology? How do drugs interfere with the disease pathway? And how can I test the efficacy of proposed drugs? During this week's live session, we're going to be looking at clinical trials. That is going to be the method that drugs are used to see whether or not they work and whether or not they effectively treat the disease compared to a placebo or something that isn't meant to treat the disease. And so we're really going to be looking at how we test our new technologies using the scientific method and how we can do them in a systematic way. Attendance at this week's live session is really going to prepare you for a project that we have later this week called pharmaceutical development. And that's going to be an individual project in which you tell us about a disease pathway and develop a drug in order to treat the disease or its symptoms. So we have a couple of key assignments for this week. You're going to participate in a discussion of the ethical issues surrounding CRISPR, which is a technology that we can use to edit genes. We're going to analyze how we can manipulate biological processes of bacteria to benefit human outcomes. We're going to identify the usefulness of various biotechnology tools. And finally, we're going to analyze a disease pathway and develop a drug to treat that disease. And that's going to be what you do in that pharmaceutical development project that I mentioned that corresponds to the live session. This week, you have a couple of resources that are really going to be helpful. The weekly agenda and pacing guide will keep you on track during this hectic week. The weekly checklist is going to help you at the end of the week to make sure that you've completed all of your assignments, but also to hear back from me whether or not I received all of your assignments and whether or not I was able to enter them and give you some good feedback. And finally, you can always email me at catherine.masick@tiponline.duke.edu with any questions. Keep a lookout for my class announcements, and I hope to see you in the live sessions. Have a great week.